So I'm interested in marine diseases, when and why they occur, how they impact populations and communities, and how and when we should manage them. Infectious diseases are a result of an interaction between a host, a pathogen, and the environment. The host in the ocean could be any marine plant, animal, or other organism. And, it could be a variety of different pathogens: virus, bacteria, fungi, protist, or macroparasite like a sea louse. And we're interested in a variety of environmental conditions. As you all know, there's many shifting environmental conditions in our oceans. When we have the right combination of host, pathogen, and environment, we get the emergence of disease. And these diseases can, in some cases, have pretty profound impacts on populations and communities. So I'm illustrating a few examples here. At D is a coral disease, which often tends to be dysbiotic and sort of potentially a number of different microbes interacting, and has had profound impacts coral diseases have on this ecosystem engineer and the habitat created by corals, and is very much impacted by temperature. B shows a picture of eelgrass wasting disease, which sometimes seems to be fairly endemic and have very few consequences for eelgrass meadows. In other cases, has been associated with really profound declines, particularly in the 1930s, when about a 90% loss of all eelgrass in the North Atlantic occurred, and it has been associated with this disease, although they did not have the methods we have now to really tease out cause. F shows a lobster with a really severe epizootic shell disease. This is also a dysbiosis, so the whole microbiome on the exoskeleton of the lobster is actually eating away at the chitin. And this community of bacteria that are found on the lobster exoskeleton actually changes as the disease progresses. So it's a shifting community, which makes it really challenging and also interesting to study. And in A, I'm showing a picture of sea star wasting, which we actually don't know if it's associated yet with a pathogen or not, but it has been associated with environmental conditions, seawater warming, and has had profound impacts on more than 20 species of sea stars. So I picked these species because they're important for habitat or for fisheries. We've got ecosystem engineers and keystone species here. And so you can imagine that disease not only impacts the species that's infected, but can have many knock-on effects as well, both ecologically and socioeconomically. One other factor I'm really interested in is fishing. I think there's lots of different human and anthropogenic actions that can alter disease, but fishing in particular can impact population sizes. It can impact fish behavior, and it can target specific individuals that may or may not have disease, and this can have really important impacts for epidemics. And in some ways, it means that we're already so somewhat indirectly, by the act of fishing, we're managing a disease, but sort of not with a strategy. So I think that there's a lot of opportunity to use fisheries for disease management, and also to use data collected for fisheries management to understand disease processes. So this is a figure that Chelsea Wood, who's a fantastic disease ecologist, made, that just sort of walks through all the different ways that fishing can impact parasite or pathogen abundance. So you can either remove parasites. Hopefully you can see my cursor now. You can remove parasites or you can add parasites, and it can have either population level or community level impacts. And it really depends on on the fishery itself, whether it's targeting a host, infected hosts, specifically uninfected hosts, or other members of the community, like the prey or the predators. And so the take-home message is that depending on the fishery and how it acts on a population, you can have all kinds of different predictions you might make about how it could alter the epidemiology of a specific disease. So with all of that in mind, what I'd like to do today is talk about some of the data requirements and analytical approaches for understanding disease in fisheries. And then I'd like to present somewhat quickly 2 case studies. One is ichthyophoniasis in Pacific herring, which is this figure up on the top. It causes this sort of sandpapery pattern on the scales of this herring here. And this is one of the most common marine diseases. Affects many different species. And then the second case study might resonate with you all in the Chesapeake, because that's where we're seeing a lot of it, and that's mycobacteriosis in striped bass. And these case studies will emphasize different types of data, either surveillance data or mark recapture data, and how they can be used to make inference about different processes. And then finally, I wanted to just talk about kind of what I think are the next steps for marine disease management and sort of how to keep moving this field forward. Over time, studies of disease and reports of disease have changed. So this is a really great paper by Alison Tracy, where she looked at reports of diseases and sort of controlled for it by weighting the number of reports by the number of studies of these organisms overall. And she found that for groups like corals and urchins and mammals, or I guess mostly corals and urchins actually, there's this increase in disease reports over time, and it's very much correlated with increasing seawater temperatures in these locations. But when you look at fish species, you get for the most part, with decapods, sharks and fishes, the opposite pattern, where there's fewer diseases in fish taxa over time. And this is presumably because these organisms are being fished out, their densities are lower, and so some of their pathogens are removed, and opportunities for transmission are removed. What this study doesn't look at is the impact that the diseases that we do see in fish taxa have. And I think this is really important because we're seeing a lot of fisheries that are pretty critically impacted by disease. Here are just a few. So the Prince Edward Island oyster fishery, they've just started detecting MSX, which causes dermo disease, or am I mixing this up? Anyway, they're finding MSX, which impacts oysters, and they just found it this year, and they're already calling it a crisis with really profound effects on that fishery. In the Yukon River, salmon are really declining. They've put a moratorium on the Chinook fishery, which impacts a lot of subsistence fishermen. And one of the causes of these declines is increased water temperature that causes a disease, the same disease I'll be talking about in Pacific herring, ichthyophoniasis. And they just get horrible systemic infections, and they often don't make it up the river to spawn. And then finally, this was in the news a lot this past year, the collapse of the Alaskan snow crab fishery. There were numerous factors, including warming, but there was a disease called hematidinium, which was very prevalent at the time of the collapse and has been associated and hypothesized to have contributed. So really dramatic changes with human impacts to humans and fisheries from these diseases. So how do we measure and quantify drivers and impacts of disease in the ocean? This can be really challenging. Disease is often hard to observe. We don't always get these obvious lesions that are very visual. Sometimes we need more complex molecular tools that can be very expensive. Mortality is hard to observe. Dead animals don't stay in the ocean for long. They're often eaten quickly. We have very complex processes that distribute pathogens in the ocean. I recently read that pathogens in the ocean compared to terrestrial pathogens, they're dispersed about 2 orders of magnitude further on average, just because they can survive in the ocean much longer and are moved through the water column and the currents. And we also have a lot of knowledge gaps in host and pathogen life cycles, which can really limit how we sample and how successful we are at sampling and understanding some of the risks that these organisms are exposed to. There's also low feasibility of lab or field studies, depending on the risk. If you do a lab study, you might need a quarantine facility, which can be challenging to run, and field studies, especially for remote organisms like snow crab, can be very expensive, and so you're somewhat limited in what data you can collect. So giving you all the reasons why this is hard to do, but it doesn't mean it's impossible. The method that I am really excited about is really working with the fisheries managers to use the population data they're already collecting to understand population dynamics, combine that with disease surveillance data, which can tell us the proportion of infected individuals over time and potentially other information like how severe the disease is within an individual, and then environmental data. And when we combine this, I think we can create some models that can give us a lot of inference about causes and impacts. So I'm going to dive into these 3 data sources just a bit more. The climate data, your oceanographers, you know this all better than me, but some of the factors that I've looked at, temperature is by far the one that we look at the most, in part because it's the data we have, but it does seem to be a driver in just about every disease I've ever looked at in the ocean. Also ocean acidification, heat waves, storm frequency, hypoxia, changes in salinity, especially with glacial melt at northern latitudes. And we have a lot of more access to these kinds of data through different types of hydrodynamic models. And it just seems like they're just adding more information, higher resolution, so that we can go back and look at data sets and look at hindcast data or forecasted data, and with higher and higher resolution, and learn more from these by reanalyzing the data in the context of this higher resolution environmental information. So I always tell aspiring disease ecologists to make friends with an oceanographer, so you can understand this data, but since you're oceanographers, make friends with a disease ecologist. So it's a great way to collaborate. So thinking about your climate data, questions are, should you look at anomalies or actual values? Do you expect to see a linear effect? We often see these sort of performance curves where you see an increasing performance with temperature and then a rapid decline once you pass some temperature threshold. And often we see diseases occurring right around this threshold when organisms might be immunosuppressed, or they're just kind of just with climate change, they're starting to live at the edge of their temperature threshold. So we might actually expect some nonlinear responses to disease or to temperature, some nonlinear disease responses to temperature. So it's just an important consideration. When we think about the disease data we collect, we can do surveillances where we just look at what proportion of the population is infected. We can piggyback this on fisheries monitoring. That's a very effective way to get data. in a less expensive way if you can get on the same boat or add it to a monitoring protocol. This tends to be lethal collections. If you're lucky enough to have a disease where you can actually look at it and do a diagnostic without killing an organism, you might be able to do a multi-state mark recapture. And I'm hoping to demonstrate just how powerful this approach is. These three pictures are all pictures of organisms that have been studied with multi-state mark recaptures to understand the impact of disease. And the reason we could do this is because for each of these organisms, we can identify whether or not it's diseased just by looking at it. So in the top, these are snow crab, and the ones on the left in column A and B, they kind of have a cooked appearance. It's a very obvious indicator that they are diseased. So we can tag infected and uninfected, or at least healthy appearing crab. We don't actually know if they're truly uninfected. And look at recapture rates, and those relative rates can tell us something about survival. This striped bass has very obvious lesions from mycobacteriosis, and then the bottom picture is a lobster with epizootic shell disease, which also has obvious lesions. The challenge with the multi-state mark recapture is that you have to be out often enough to get enough recaptures that you can actually do an analysis and have enough statistical power. Then finally, there's various hybrid approaches where you can combine lab and field data, maybe inform mathematical models, and make inferences that way. So the first case study I want to present is using multi-year surveillance data. So in this case, we're looking at Pacific herring, and they get these ichthyophonus infections, which cause systemic infections, but we tend to see the highest level of infection in the heart, and you can see the histology here. The pathogen is these bright purple circles, and then in some cases you can see these granulomas surrounding the circles, just the sort of dark, dense tissue, and that's the host just trying to wall off the pathogen so it doesn't spread. And those granulomas are thought to actually be as damaging as the infection itself. So we see very high prevalence of this disease in Gulf of Alaska herring, between 15 and 40%. And it varies with location. There's no known recovery of herring. So once they're infected, they will eventually die, but we don't really know how quickly they die. And we actually see very infected herring. This one over here that's sort of partially dissected, you can see the heart is mostly white because it's so infected, but this herring was still swimming. So they're able to survive very intense infections. Because of their role as forage fish and their important role in the food web, it's really important to understand how this disease is impacting populations. So if we want to make a very simple model of this disease, we would call it an SI model. So we have susceptible herring, and then they can become infected and they never recover. So they're either susceptible or infected, and once they're infected, they eventually die. So we have death due to natural mortality and then disease mortality, and a small equation that dictates the rate of transmission. It's a very, very simple disease model. And we can ask, well, what causes this population variation in prevalence? And for this study, we had access to 2 locations, Prince William Sound here and Sitka Sound. They both have historically had really large herring populations, but the population in Prince William Sound has declined considerably since the Exxon Valdez oil spill. So if we're interested ultimately in these demographic and environmental correlates of disease, we kind of have to take steps back. Like, we want to understand the age-based prevalence. We want to understand the age structure of the population. We want to know what the population size is. There's this kind of all these things that go into actually estimating an accurate estimate of disease in this population, and it requires data from a number of sources. So for this study, we actually had 6 different sources of data. We had our surveillance data, which told us about how much disease was in the population. We had population data, which was from the Alaska Fish and Game surveys they do for the stock assessment. And then we had all of this environmental data about upwelling, the Pacific Decadal Oscillation, North Pacific Gyre Oscillation, which from talking to fisheries managers, we knew these were important for dictating herring abundance. And then we had estimates of herring abundance and recruitment, which came out of the stock assessment models. So we put all this together and we were able to create this age-based, sort of age-weighted estimate of disease prevalence for our 2 study sites, which looked like this. So we had data since 2007, and for Prince William Sound, the prevalence was about 40%, a little over 40% at the beginning of the study, and it declined over time to a little less than 20%, but it was almost always higher than Sitka Sound. And Sitka showed a similar declining trend in prevalence. So we have this data. The data, the surveillance data coincided with the stock assessment. So once a year, when they're collecting data to figure out how big the population is, we just would take a subsample of those herring and do necropsies on them and determine the infection status. And so that's how that data was collected. So Prince William Sound has consistently higher levels of disease, but we found that the severity of disease was not greater. So what we did here was we looked at what proportion of those infected herring had disease that looked like this, where we could actually see it histologically. Sometimes we just detect the pathogen, but there's no evidence of actual damage to the heart. So we considered those damaged ones to be what we called high occupancy infections. And you can see the estimate for that proportion of high occupancy infections was pretty much identical for the 2 populations. We then looked at age relationships for these populations. So Prince William Sound is at the top and Sitka Sound is below. And each of these lines is following a cohort of herring over time. So you see, for example, in green here, this is the cohort when they're 3 years old, 4 years old, 5 years old, 6, 7. And you can see in most years, you have this positive relationship. As the fish gets older, they're more likely to have disease. So maybe that could explain that age relationship is what could explain the higher disease prevalence in Prince William Sound. Maybe those fish are just older, and so they're more likely to be diseased. That's a reasonable hypothesis. But if we look at the age structure of the population, again, this is Prince William Sound in pink here, you can see that the estimated prevalence sorry, this is age here. I think I cut off the x-axis. So this is age of the fish, and this is the estimated prevalence on the y. We are estimating that these fish have higher prevalence in Prince William Sound for the same age. So it's not explained by age. So then we did some generalized linear models to see if there were environmental variables that could explain these population level differences. And we found that disease tended to be associated with negative values of the Pacific Decadal Oscillation, but it was true in the same way for both populations. So it's explaining the trends across these 2 populations, but it doesn't explain the site level differences. So to summarize, we see that age and temperature, which is associated with the Pacific Decadal Oscillation, are driving some of the dynamics of this disease, but they don't explain why we're seeing differences in these infection levels. And it's important to understand this because this Prince William Sound population used to be a really important fishery, and it has collapsed and not recovered for over 20 years following the Exxon Valdez spill. And so we think that this disease might be the reason that this population has failed to recover. So potential hypotheses as to why we see these higher infection levels is maybe there's increased exposure to the pathogen that causes this disease, Ichthyophonus, in Prince William Sound, or there's increased mortality. I meant to write Sitka Sound here, I'm sorry. Increased mortality of diseased fish in Sitka Sound. So you get they're all exposed to the same amount of disease, but you get a lot more mortality, so you get a lower prevalence. One of the challenges of understanding this particular disease is that we don't really know how the pathogen is transmitted. So without understanding the mechanism for transmission, it's really hard to understand why there might be more or less exposure. To get at this question a little bit more, we used an approach called a partially observed Markov process model. And it sounds very complicated, but to kind of break it down, partially observed is just referring to the fact that we're just observing a subset of a population, that subset reflects the whole population. And so that could be the 60 fish we sample every year, or the small amount of fish that are collected to estimate the population size. It's a partial observation. And then a Markov process just means that every time step is explained by the previous time step. So we have basically a latent process here, which is sort of our disease model, the susceptible infected model that I presented, an observation process, which is how we sample our data, and then the data that come out of that observation process. So we can use this to actually estimate the mortality and the transmission rate of this disease, even though we don't actually know the mechanism for transmission. So here we're trying to quantify exposure to the disease, transmission, and mortality using this partially observed Markov process model. So the model here should be familiar. Here's our susceptible infected model that I presented earlier. And if we add our susceptible and infected together, we get the total population size. And we have some kind of recruitment. So every year we have susceptibles recruiting into the population and infected individuals recruiting. And the difference between the number that are susceptible and infected should be reflected from our pathogen surveillance data. So again, we've got our data, our observation process, and then the model that it's feeding into. And finally, our total population size is estimated from these acoustic data surveys. So we're taking a whole bunch of data and seeing how it fits into this process model that we think is dictating disease in this population. And we run multiple versions of this model and use log likelihood to find the best model, which basically maximizes the fit of the model, which is in red here, to the data, which is in blue. And we get, so that these are these ones with the highest log likelihoods. And we have an estimate of a transmission rate of 16% and a mortality rate of 20%. So this is a way that we can start to get at those questions I couldn't answer with the surveillance data, but it really takes understanding at least some of the biology to have a decent model to put into this partially observed Markov process approach. So to summarize, we have the surveillance data. We can use these linear models to tell us how some prediction and some correlation between different variables, but they don't really get at the mechanism. But if we add what we know about the biology of the disease, you can start estimating these really important parameters that are mechanistic using simulations and approaches like the partially observed Markov process model. the caveat is lots of different data types might be required, and it really requires talking to resource managers and oceanographers and various people to understand what's out there and really get an understanding of both the biology and the environment. So you are creating a model that it makes sense given the knowledge there. Hopefully this isn't too quantitative or too fast, but I have one more example I'd like to share. And this is a different approach. This is the mark recapture approach, and we call it a multi-state mark recapture because we're not just tagging and recapturing individuals, we're tagging them and then looking at the state of their health. So we can say this one is obviously diseased, and we can have others that are obviously healthy, and we can rank them in terms of mild, moderate, or severely diseased. And the question here first is, can we estimate the mortality rates of these fish and the transmission rates of the disease? And then can we find any environmental correlates? And so in this case, we used data from a ROMS model, an oceanographic model of the Chesapeake Bay. And we were interested in both temperature and hypoxia. And specifically, if the hypoxia that happens in Chesapeake Bay is forcing these fish into warm surface water where they're going to be more likely to get disease. This Chesapeake Bay is a really important nursery ground for striped bass. And this disease has not been here for that long, just a couple of decades. And it seems to be getting worse, and we're seeing declines in striped bass. So understanding what is driving this, why this disease is here, is really critical for management. It's also a really important recreational fishery and important just for tourism and kind of a way of life in the Chesapeake. So we can make a disease model for mycobacteriosis. It's exactly the same. This is very similar to ichthyopaniasis in the sense that we have animals that are susceptible, they get infected, there's no recovery. It's a very slow progressing disease. So we have animals that can live for years with infection, but they do eventually die. We don't tend to see big mortality events like we might see if it was a fast-acting disease. So similar model, but very different approach to understanding disease mortality and transmission. So in the multi-state mark recapture approach, the analytical approach estimates the survival of organisms, transitions between states, so transitions between healthy and diseased. It estimates capture rates. And for all of these estimates, you can add individual covariates like the size or the sex of the individual, or environmental covariates like the temperature and hypoxia. And then you run a number of models and use model selection to find the best fitting model to your data. There's two types of software that can do this type of analysis. There's one is called eSurge and the other is called MARC. So for this analysis, we used eSurge. Here you can see the 4 different stages of disease from A to D, or healthy to severe disease. And we had to specify a matrix that basically shows transitions between one time and the next time step. And that matrix is used to, one is for specifying survival, one is for transitions between stages, and one is for capture probabilities. I'm not going to get too much into the modeling, but it's pretty neat. So skipping ahead to the results, we were able to estimate, again, this was yearly, we got yearly estimates, although the sampling occurred more than yearly because we needed recaptures for these fish. So we were able to estimate incidence rates. So these rates go from 0 to 1 or 100%. So you can see 60% healthy fish become moderately infected every year. It's a really high rate. These were just adult fish, so we don't have estimates for juveniles. And then we also found that both temperature and disease affect the survival of striped bass. So we also didn't find any evidence that hypoxia was a contributor, but during the study years, there just wasn't that much hypoxia, so it wasn't a really good test of that hypothesis. But what you can see here is the estimated model predicted yearly survival for healthy, mild, moderately, and severely infected or severely diseased striped bass. And then below is this average summer sea surface temperature. And you can see that the years where you have really high sea surface temperatures, the survival is very low, almost zero for severely infected fish, but it's still fairly low for healthy fish as well. So it's not just the disease that is killing these fish. They're really living above their thermal threshold. And then the other interesting thing we found was that the capture probability, so the likelihood that a fish would end up in the gill nets that were used to catch them, was incredibly high for the diseased striped bass. It was about 8 times higher than it was for a healthy striped bass, which means that as a surveillance tool, this is fantastic. If there's a diseased fish, it's very likely to end up in this net. But if you just look at the proportions of healthy and diseased fish in your net, you're going to overestimate the disease prevalence. So you have to kind of back calculate to take into account the bias in this approach. We can also take these data or take the parameters from this model and create a mathematical model to look at the cohort survival over time. So in this case, we're taking 10,000 fish, 10,000 striped bass, and we're just projecting their population, not adding any individuals, but just what is the fate of these 10,000 fish over time at 26 degrees, 27.5, and 29 degrees Celsius. And you can see the impact of temperature is incredibly dramatic. You're not going to get fish surviving to reproduce at 29 degrees Celsius. And even at 26, your population is going to be very diseased. And that really matches the observations that we had. So to summarize this approach, it's a really powerful tool to estimate these epidemiological rates like survival and transmission. You can easily incorporate and test for environmental drivers. It can be quite expensive. Typically, you either need to offer a reward for somebody getting a tagged fish. So $20 or $100 if they get a fish and send it to you so you can evaluate it, or you go pick it up and they freeze it so it doesn't further degrade. Or you just need boats out recapturing them. And then finally, you need this non-lethal diagnostic. So you can actually assess the disease state over time. So this is not appropriate for all fish. We didn't use this for herring because you will never recapture your herring. But for a fish like striped bass, the recapture rates were about 25%. So that was good enough. And we had enough tag that we could do pretty well with this. So to conclude about these quantitative approaches, there's a lot to consider. Your environmental drivers, like the variation, the time frame that's relevant, how you summarize them. Are you looking at thresholds, or are you looking at linear or curvilinear processes? How do they relate to host physiology or pathogen physiology? You need to know your biology and define your disease model. Is it susceptible infected model? Do you have recovered individuals, which we call a susceptible infected recovered model? Do you want to model the pathogen itself? This would be a susceptible infected pathogen or particle model. So you have to make sure your model reflects the biology, and you want to leverage multiple types of data sets. In my case, this means I need to befriend an oceanographer and probably a resource manager. And then collaboratively, we can combine this data to create a good model. And then finally, I think there's just very powerful underutilized analytical tools to estimate these processes. And the ones I presented are all adapted from human disease models. So really paying attention to what they're doing in that world and seeing what we can do to use them for marine processes. So just have a few minutes left. I just wanted to think about how this work informs management. I was part of a working group that we wrote a paper together on marine disease management, and we came up with a couple ideas, which I'll present. One is thinking about sort of the ongoing things that have to happen to understand disease, like surveillance, basic and implied research to understand drivers and outcomes of disease, like mortality, and then ongoing communication with stakeholders, researchers, managers, so that if there's some kind of management approach that's needed, there's already trust built so that people will understand and why you want your commitment to the project and why the work is necessary or the management tool is necessary, especially if it's something like a fishing moratorium. And then you have reactive processes like mitigation. If you have an epidemic, you can mitigate the direct impacts of the disease, which might be ways of culling infected individuals, ways that you can impact the population that's infected, or you can mitigate the downstream impacts. So this could be something like helping fishermen find an alternate, pushing them towards an alternate source of income, or building resilience in a fishing community so that if there is disease in the main fishery, there's something else that they can fish. Typically, a disease epidemic looks something like this. Without management, you might not detect the disease until the epidemic is at or past its peak. And that leaves very little opportunity for any mitigation effects, processes. If you have a surveillance program, you might detect the disease earlier and be able to do some implementation and mitigation processes. And if we know enough about the disease, we may be able to do some forecasting and modeling and predict when we think it should occur. And in this case, we can kind of flatten the curve the way we've talked about. You've probably heard with COVID. You can actually, maybe you can do more culling. You can intervene early enough that you can have more of an effect. Practically, there aren't very many examples where fisheries have been managed for diseases. A few examples are here. The first is with snow crab that have hematidinium or bitter crab disease. Again, these are the ones that kind of look cooked. On the left side, both of these figures. And in this case, in Newfoundland, the regulation was that you can't throw these crab overboard. They need to be discarded on shore so that they don't spread hematidinium. Another management tool has been with viral hemorrhagic septicemia, which causes hemorrhaging in numerous species of fish, including herring. This is a young Pacific herring that has, you can see hemorrhaging around the eye and the mouth and the fins. There's a fishery called the spawn-on kelp fishery for herring. And that fishery, basically, when the fish are about to spawn, they're put in an open net impoundment. So the net's in the ocean, there's water flowing in and out, and they put kelp, these strings of kelp, and the fish spawn on the kelp, and then the kelp can be harvested, and then they let the fish go. And they're only in the impoundment for about a week. It seems very sustainable, except that the stress of this impoundment often causes the fish to have an outbreak of this very fast-acting disease called viral hemorrhagic septicemia, which can be highly lethal. And the act of spawning attracts other fish to the net. And so it creates, there's spillover into fish that are outside of the net. So this closure of this particular fishery has been effective. And then here's another example with the Yukon River salmon that are getting ichthyophonus, which you can see kind of in this, light colored, there's kind of speckling in the heart of this Chinook here. That's been associated with mortality of these fish before they are able to spawn. Initially, there were changes to the timing of the fishery so that fish were caught before they were likely, before the outbreak occurred. And now there's a 7-year moratorium on the subsistence Chinook fishery, which has huge impacts for native Alaskans that live up there and rely on this fishery, but is at this point considered the only way to help the fishery recover. I'm running out of time because I want to make sure there's time for questions. One final approach is to directly incorporate disease into a fishery's a stock assessment. This is typically done by just using the prevalence of the disease or the proportion of individuals that are diseased as a covariate in the stock assessment. And that's problematic because if the individuals are sampled in the early part of the epidemic, in the middle, or at the end, the prevalence is going to vary. And so they're not really it doesn't necessarily indicate what the mortality is due to disease. So in this case, we have a paper led by John Trachta where we had a dynamic model of the disease that was incorporated into a stock assessment model. So it's not just modeling one level of disease, it's actually modeling the whole process that would result in the prevalence of disease that's seen. So I'm going to skip going over the details of this for the sake of time, but feel free to ask questions or I can point you towards the paper. And my hope is that this type of approach might be able to be adopted for certain fisheries. Just going to skip over this example. So just to kind of summarize how this disease work can inform management, we can inform surveillance methods so that there's targeted surveillance to see a disease when it's occurring and have an early detection of an outbreak, which can lead to an early response. We can potentially forecast future disease risks. We potentially can add disease to stock assessments, so it's managed within the same tool that's being used to manage a fishery. I think with the increasing environmental and oceanographic models, we can use existing fisheries data to answer new questions about disease. And I think we can also use these data and models to develop communication tools where we project different scenarios and hopefully can increase understanding of the importance of this type of work. Just going to quickly advertise that I am going to be leading a 3-week workshop in June of next year at the Friday Harbor Labs. It's really for grad students, postdocs, talented undergrads, looking at disease ecology in a changing ocean. Eileen will be a guest lecturer and we have scholarships available. So please reach out if you're interested or know someone who might be interested. And if you're really excited about this topic, we also have a special issue in Philosophical Transactions of the Royal Society on managing infectious marine diseases in wild populations coming out in about a year, I think, hopefully. So lots of people collaborated on all this work. Lots of fisheries managers, oceanographers, and really it's just a huge collaborative effort to try to understand these processes. So thanks to all those folks, and I would be happy to take any questions.
Okay. Well, thank you for a really interesting seminar. You do have one question in the chat from Robin Dunbar, asking if there are health risks to the watermen if they handle the diseased fish.
That's a great question. So most diseases that we see in fish and shellfish cannot be transmitted to humans. But there are a few exceptions, and the mycobacteriosis in the striped bass is one that actually can be transmitted. Sometimes it's called fish handler's disease, and it can cause, they almost look like warts, like small lesions that don't heal. And for folks that do get infected, it can take 9 months or a year on antibiotics to recover from those infections. Mycobacteria is the same, in the same class of bacteria that causes tuberculosis, which is also something, very, very hard to get rid of, lots of antibiotics, very persistent infection.
That's interesting. I never knew that. I guess I had thought most of these diseases were not transmittable to humans, but it doesn't sound good.
Yeah, most of them aren't.
Are there any other questions? If you have a question, please just turn on your camera and ask the question. Yeah. John, did you have a question?
I had a quick one, kind of Maya, thanks for that. Kind of a philosophical, technical question is that a lot of the SI models are based on human diseases and contact. So the transmission rate of beta times S times I is based on frequency of people actually getting in the vicinity of each other. Marine diseases seem to have this long range transmission process from, MSX and so forth and so on. How well does that simple S times I model seem to work, or should we modify that to take advantage of the fact that marine diseases tend to spread rather broadly?
That's a great question, John. I don't know that I have a good answer. I think the hydrodynamic models that actually model pathogens dispersing, they can incorporate the biology of the pathogen and how long they persist at water, that can sort of get at like how important is this long distance transmission and where our sources and sinks. But I guess depending on your system and, for something like herring, we don't really have a sense of like how much they're moving around and what's happening when they're at depth. And there's so much we don't know about their movement and the pathogen movement. So I guess sticking with a simple model is kind of a good starting point. Sometimes I'll use, I can go back to it. There's a coefficient you can use that sort of lets you estimate the relative amount of frequency dependence that's happening with a disease. Oh, sorry, it's going to take me a minute it's here, this N to the Q.
Right.
So if you have enough power in your model, you can maybe estimate like the relative frequency or density dependence of the transmission, but
that sort of makes the beta parameter density dependent in some way, if you want to think of it that way. Well, I guess my question, I'm not surprised by your answer. I guess my question really was, does this model seem to work acceptably well given what you know and what you can represent? And I'm sort of hearing you say it seems to do okay, or it doesn't do poorly, so we'll stick with it. And I agree with you, simpler is better than being exotic at this point if you don't really have details to make the exotic model better.
Yeah. I mean, I don't know if we can tell just how well the model works. I mean, I guess the here, you can see all these different model runs and the data we're matching it to. And you know, there's areas where it's not performing well. But, you know, there's also a lot about the biology of these fish that is just assumed, you know, the way we're estimating population size from acoustic data. And so there's so much noise. Sometimes I'm just impressed we can get some signal out of it. So
One other very quick question. I know there's another comment in the chat. I noticed in the striped bass results that there didn't seem to be a temperature effect on the infection. Sort of looking quickly at the figures you showed, at higher temperatures, all disease states seemed to die at the same rate, and it didn't seem that warmer temperatures led to more disease, which led to more death. And I don't know if that's a way to look at it.
yeah, I think we found no effect of disease on, no effect of temperature on
Temperature on disease
on,
state.
on incidence,
Right.
like the rate of new infections, but the probability that you survive the infection is lower at higher temperatures.
Okay, thank you. I'll stop at that point.
Okay.
And maybe for our last question here, we have one from Cynthia Jones, following up on John's question. Is there a component related to the migratory scope and stock mixing?
At this point, I think that's a sort of a future direction, you know, once we understand if we can understand migration patterns and, you know, if we could put temperature sensors on these fish and then really understand what they're exposed to in terms of temperature, for the stripe, we tend to use, like, for these two models, sort of really regional estimates of temperature, because we don't know kind of within a basin, are they going to really cold deep water? Are they staying high? Are they, you know, maybe they're moving to get out of disease conducive temperatures. And I think there's a lot of really fascinating behavioral work to do as in relation to that. But it's yeah, big future direction, I guess.